Now we're going to start exploring the diversity of life by looking at the different domains and kingdoms, starting with our prokaryotic groups, the bacteria and the archaea. Bacteria and archaea are considered microbes. A microbe is any living thing that is too small to see with the naked eye. Microbes are not all closely related to each other. There are microbes in all three domains, meaning microbes can be either prokaryotes or eukaryotes. Even though viruses are not even considered living, they are also often referred to as microbes. While they are individually small, the bacteria domain has incredible diversity and abundance. Just 1 teaspoon of soil may contain as many as 1 billion bacterial cells. If we compare bacteria to other forms of life by looking at their total biomass, we can see that they make up a large portion of the weight of living things on Earth. If we combined all the bacteria on Earth and weighed them, they would weigh more than all animals combined. We can use the shape of bacteria to help us identify them. Kochi are bacteria with a spherical or round shape. Bacilli are rod-shaped bacteria. And spirilla are long and thin with a squiggly or spiral shape, like a piece of spaghetti. If you haven't already, pause this video and open up Participation Assignment 3 and answer these 2 questions. Bacteria reproduce asexually by cloning themselves. As they reproduce, they form colonies. In addition to the shape of bacterial cells, we can also use the visual characteristics of the colony to help us identify them. Colonies of different species may have different shapes or colors. Bacteria usually reproduce very quickly. 1 generation for a bacterium may be as short as only an hour. Bacteria are prokaryotic cells, so they have all the characteristics of prokaryotes. In addition to a plasma membrane, most bacteria also have a cell wall. Together, the plasma membrane and the cell wall are known as the cell envelope. Like all prokaryotes, the fluid and other structures inside the cell are known as the cytoplasm. Like all living things, bacteria have genetic material that together is known as the genome. The genome is composed of DNA. Like all cells, bacteria also have ribosomes, which are the structures that make proteins. Some bacteria also have an outer coating called a capsule that helps hold in water. Like in all prokaryotes, the genetic material of bacteria is circular and is found floating in the cytoplasm rather than contained in a nucleus. Bacteria can have DNA in 2 different structures. Most of their DNA is contained in the chromosomes. This DNA encodes all of their basic life processes. However, bacteria can also have additional DNA in plasmids. The plasmids contain the code for some specific functions. Different strains of bacteria, even in the same species, can have different plasmids. When we think of E. coli, we often think of the strain that makes us sick. However, we all have E. coli living in our digestive system right now, and they don't cause us any harm. However, some strains of E. coli contain a plasmid that magnifies the gene for a toxin produced by E. coli. It is the presence of this plasmid that makes these strains of E. coli virulent or disease-causing. Here are 2 more participation questions for you. Bacteria reproduce asexually, which means they make genetically identical copies of themselves. The process by which they reproduce is called binary fission. Binary fission happens in 2 steps. In the first step, the bacterium replicates its DNA, both the chromosomal and the plasmid DNA. Then the cell elongates and divides into 2 daughter cells, each containing the same chromosomal and plasmid DNA. Because bacteria can only reproduce asexually, they are not exchanging genetic material with each other when they reproduce. Instead, the daughter cells are clones of the original parent cell. But they have evolved some creative ways to increase their genetic diversity. Here's another participation question for you. Make sure your spelling is correct for full points. Capitalization doesn't matter. Bacteria have evolved some creative ways to increase their genetic diversity by exchanging genetic material with one another without reproducing. This would be like you and your friends just sharing some of your genes with each other. In conjugation, a donor bacterium transfers some or all of its genetic material to a recipient bacterium. In transformation, a bacteria takes up genetic material from its surroundings. This might be from a bacterium that has died and split open, leaking its DNA into the environment. Transduction happens when a bacterium is infected by a virus that contains some DNA from its previous host. When the virus injects its own genetic material into the bacterium, it may also introduce some foreign bacterial DNA, which can be taken up by the recipient bacterium. Remember, this is not reproduction. In all these forms of lateral gene transfer, bacteria are sharing DNA with others in the same generation. Now test your knowledge on how bacteria exchange genetic information with these 2 participation questions. Remember, bacteria are an incredibly diverse group of organisms. They have evolved to digest and obtain energy from many different food sources. There are bacteria out there that can digest almost anything, even some who can digest plastics. Scientists are now exploring ways to bioengineer bacteria to break down plastics and other waste products. We can organize bacteria into groups based on the ways they obtain energy. Any living thing that needs to eat organic molecules to get energy is known as a heterotroph. Heterotrophic bacteria are known as chemoorganotrophs. Chemoorganotrophs digest organic molecules. If you haven't taken chemistry, an organic molecule is any molecule composed of a carbon backbone. Notice that orgo is right in the name. This can help you remember that chemoorganotrophs eat organic molecules. Organisms that get their energy from either sunlight or inorganic molecules are known as autotrophs. Chemolithotrophs digest inorganic molecules, often minerals. Litho means rock, so these bacteria are sometimes also referred to as rock eaters. Photoautotrophs use sunlight to convert carbon dioxide to glucose, just like in photosynthesis in plants. Now pause the video to answer this participation question. When we think of bacteria, we often think of the ones that make us sick. However, most bacteria are harmless to humans, and many are actually beneficial. For example, we rely on bacteria to produce some of our favorite foods, like yogurt and Cheez-Its. While beer primarily relies on fermentation by yeast, some beers are also made using bacteria. We are just starting to discover all of the incredible benefits we get from the beneficial bacteria living in our digestive system. In some cases, they help us digest certain foods. Our digestive system is an ecosystem, so the presence of helpful bacteria also limits the resources available for harmful bacteria. Other beneficial bacteria release chemicals that are important for human health and regulation of different body systems. As we mentioned before, most bacteria are harmless to humans. Only a small number are pathogenic or disease-causing. Some pathogenic bacteria are only harmful under certain conditions. We all have the bacterium that causes strep throat in our noses and mouth right now. Most of the time, it is completely harmless. But remember how we said that the presence of helpful bacteria can keep pathogenic bacteria in check? When the helpful bacteria in our nose and mouth are disturbed, the strep throat causing bacterium can start to take over. This is what leads to strep throat.